everyone, and welcome to Miss Estruch Biology and this entire topic 3 theory video. If you want to be able to make notes as you go in a structured way, then check out my free workbook in the description, which is designed for you to fill in the information from each slide as you go. But if you want ready-made notes with the key marking points, exact mark scheme answers, plus examiner's tips, then don't forget to check out my completed A-level notes or my flashcards. Enjoy the video, everyone. So topic 3 for AQA A-level Biology, and we start with 3.3.1 surface area to volume ratio. Exchange surfaces in organisms, so where you might have diffusion occurring across, for example, have many similar adaptations to make that transport across the surface more efficient. And surface area to volume ratio is one of those adaptations. And the relationship between the size of an organism or the structure, so like the shape of a cell or the organism, and its surface area to volume ratio plays a significant role in the types of adaptations an organism will have. And we're going to see this repeatedly throughout topic 3. So the concept of surface area to volume ratio is, if you were to work out the surface area of the object, and in the case of a cube, that would be the area of one side of the cube, so the length times the width, and then we times that by 6 because there's 6 sides to the cube. So our surface area for this cube is 6 centimeters squared. The volume would be the height times the length times the width, and 1 times 1 times 1 is 1 centimeters cubed. The surface area to volume ratio means divide the surface area by the volume. And that gives us 6, or a 6:1 ratio, but we can just present that as 6. If we then were to calculate that for these larger cubes, we can see this one, the surface area to volume ratio would be 3, and this one would be 2. And this is demonstrating a key pattern. The larger the object or the organism, so the bigger the size, the smaller its surface area is compared to its volume. And that means that small organisms, for example, an amoeba, has a very large surface area compared to its volume. And this means that there is a big enough surface for exchange to happen across to compare to its volume. But also there's a smaller distance from the outside to the very middle of the organism. So you have a short diffusion distance as well. Now, as a result of this, very small organisms don't have special exchange systems that they've evolved. For example, a lung system or respiratory system, we should call that. The lungs is the organ. And therefore, they can just simply exchange substances across their surface. So as an organism increases in size, their surface area to volume ratio gets smaller. So that means larger organisms have a relatively small surface area compared to its volume. So they don't have a very big surface area available for exchange of substances like oxygen or heat compared to their volume. Whereas in the smaller organisms, as we said, they have a large surface area to volume ratio, and that enables efficient exchange directly across their body, so they don't have to have any specialized exchange or transport systems. Now, smaller mammals have a high surface area to volume ratio, and that means that they lose heat rapidly across their surface. So to replace that heat that they lose, they have to have a high rate of metabolism, and therefore they will respire rapidly. And this high metabolic rate provides the energy needed to provide more heat and maintain a stable body temperature, especially if they're in colder environments. Larger animals, because they have a smaller surface area to volume ratio, they will lose heat more slowly due to that. And their metabolic rate can therefore be relatively lower in comparison to the smaller mammals. So larger organisms then will have adaptations to increase the surface area to volume ratio at these exchange surfaces. So for example, villi and microvilli, which we'll see come up in the digestion topic in this video. Alveoli and bronchioles, spiracles and tracheoles, and gill filaments and lamellae, and also the thin wide leaves are all linked to gas exchange. This one here is in mammals, this one is in terrestrial insects, fish and plants, and this is all going to come up in this video. And then also many capillaries. So having a capillary network is going to be relevant at exchange systems for gas exchange, but also for the absorption of digested food. So these concepts will come up over and over again in this video. 3.3.2 is gas exchange, and some key terms to be familiar with this topic are breathing. This is the movement of air into and out of the lungs. Ventilation is the scientific term for breathing. Respiration doesn't really come up in this topic too much, but I just bring it up because that's commonly mistaken for breathing at GCSE. So let's just clarify. Breathing and respiration are not the same thing. Breathing is air moving in and out. Respiration is the chemical reaction that releases energy in the form of ATP. And then gaseous exchange, that is diffusion of oxygen from the air in the alveoli into the blood, and of carbon dioxide from the blood into the air in the alveoli. Now, gas exchange just means the exchanging of gases across a surface, and that will be by diffusion. So gas exchange surfaces all have the same three features that they'll have adaptations for. Large surface area to volume ratio, short diffusion distance, maintaining a concentration gradient. And when we have a look at the gas exchange system in mammals, for example, humans, in terrestrial insects, in fish, and in leaves, we'll look at what do they have as adaptations to meet these needs. And that's the common marking points that come up for all three. And it links to this equation here, which you don't have to know, but it just helps to explain it. The rate of diffusion is proportional to how big the surface area is, how steep the concentration gradient is, divided by the length of the diffusion pathway. So if we have a look at the human gas exchange system, these are the 5 structures that you need to be familiar with. So the alveoli, which are the air sacs. We then have the bronchioles that attach to those and they're branching. You then have the bronchi, which is the 2 splitting tubes from the trachea, which is your windpipe. And the lungs, you have a left and a right lung. Ventilation is how we get air in and out of the lungs. And you need to know about the role and what happens to the diaphragm, the rib cage, and the intercostal muscles. So ventilation is inhaling and exhaling, and it is controlled by these muscles. And when it comes to the intercostal muscles, you have to be specific, talking about the external or internal. It's normally only the external that they refer to on the mark scheme. So let's have a look at these two. For inspiration, which is inhaling, the external intercostal muscles contract, and that pulls the rib cage up and out. That would be 1 mark. I've not seen on the mark scheme anything linked to the internal intercostal muscles, but just so you're aware, they'll be relaxed because they're antagonistic muscles, which means if one is contracting, the other does the opposite, so relax. The diaphragm is a muscle, so it contracts, and that then pulls it to move down and flatten. That would be a mark on the mark scheme. This then causes the air pressure in the lungs, or you could say the thoracic cavity, to decrease. And the reason the air pressure is decreased is because the lung volume is increased, or again, you could call that the thoracic cavity. Because that rib cage has moved up and out, the diaphragm has moved down, you now have a bigger space, so a larger volume, but it contains the same number of air particles. So that means that the pressure decreases. And for that reason, air moves in. So your key marking points would be this row here about the external intercostal muscles, this row about the diaphragm, and then these two combined. So that would be a 3-mark answer. Expiration or exhaling, exact opposite. So the external intercostal muscles relax, and that causes the rib cage to move down and in. The internal intercostal muscles, again, it doesn't really come up in the mark scheme that you'd have to say that they contract. So the pull the rib down and in, that would link to this mark here. The diaphragm relaxes, and that causes it to dome and move upwards. And because of that, you've now decreased the volume in the lungs or the thoracic cavity, and therefore that increases the pressure in the thoracic cavity. And because you've got a higher pressure, that is moving air out. Now, there is a maths equation linked to this, pulmonary ventilation, and that is the total volume of air that is moved into the lungs during 1 minute. So decimeter cubed is our unit for volume, per minute is how we get it as a rate. So you'd need to do the tidal volume times ventilation rate. And the tidal volume is the volume of air inhaled or exhaled in one breath or breathing cycle. Ventilation rate is the number of breaths per minute. So if we have a look at some adaptations then to increase the rate of gas exchange or diffusion of the gases, once the gases are in the alveoli, so because of that ventilation, the gas exchange occurs between the epithelium and the blood. Alveoli are the tiny air sacs at the end of the bronchioles, and in each of your lungs you have about 300 million, and that is how we get the large surface area. One alveoli does not provide a large surface area, it's the fact that there are so many in each lung. That's adaptation number one. The short diffusion distance, that is because the alveoli epithelium is just a single layer of cells. That's a very thin layer to minimize the diffusion distance. It's the same thing with the capillaries as well. It's just one layer of cells making up that epithelium. So that is our short diffusion distance. The maintaining the concentration gradient is nearly always the same adaptation, surrounded by a network of capillaries. Because what that means is, as the gas diffuses into the blood, it's then carried away and replaced by blood that is low in that gas, in this case oxygen. So those are our key adaptations for gas exchange in the human lungs. We then move on to gas exchange in fish, and fish get their oxygen from the water. The challenge they face though is there's about 30 times less oxygen dissolved in water than there is present in air. So they have to have special adaptations to really ensure that they maintain that concentration gradient even when there's so much less oxygen dissolved in water. And we'll come to that when we get to the countercurrent flow. So fish gill anatomy first then. So the gills are found on both sides of the head, and they're found as 4 layers of gills. The gills are made up of these stacks of gill filaments. So this long structure sticking out is a gill filament. Each gill filament, so we're zooming in on one gill filament here, has lamellae at right angles going all the way along it. And the lamellae is the site of gas exchange. So because you have gills on both sides of the head, you then have lots of gill filaments, and every gill filament is covered in lots of gill lamellae. That is what provides the large surface area for gas exchange. And when the fish opens its mouth, water rushes in and it goes through. There's a hole in the side of the head, which you can see here. So that causes the water to move in, goes over the gills and comes out the other side. And as the water flows over the gills, the gas exchange will happen at the gill lamellae. So other adaptations then. We've talked about how they have a large surface area to volume ratio. Next then is how is the short diffusion distance maintained or provided? And that is, first of all, because the gill lamellae are very thin, but also within every gill lamellae there's a capillary network, and the gases are diffusing from the water into the blood in the capillaries, and that's a really short diffusion distance. Finally then, it's how do they maintain that concentration and diffusion gradient across the entire lamellae, even though there's a low amount of oxygen in water. And that's when we get to the countercurrent flow mechanism. And the countercurrent flow mechanism means that the blood and the water flow in opposite directions. So this is when water flows over the gills in the opposite direction to the flow of the blood in the capillaries. That would be one of your key marking points for what is the countercurrent flow mechanism and how is it advantageous. Is that means that the equilibrium of oxygen is never reached, because as the blood flows in one direction and the water flows in the other, that means you should always have blood which has a lower concentration of oxygen reaching water which has a higher concentration of oxygen. So equilibrium is not reached. And your key marking point, your third mark would be This ensures that a diffusion gradient is maintained across the entire length of the lamellae. Because if it was concurrent flow, that should be con, not co, that would mean that the blood and water flow in the same direction, and by the time you're about halfway or 3/4 of the way across the lamellae, you would reach equilibrium, so there'd be no gas exchange or diffusion in the end section. And that's why the key mark is across the entire length of the gill lamellae. Next, then we look at terrestrial insects. They have an exoskeleton made of hard, fibrous material, and that is an advantage because it's protection and it prevents water loss, but it reduces the space that they could do gas exchange across. So they don't have lungs, and instead they have a tracheal system. And the tracheal system is made up of a trachea, tracheoles, and spiracles. Whenever you're describing this in an exam question, there's normally one mark just for stating those three structures. So always start by stating those. The spiracles are these round valve-like openings that run along the length of the abdomen, and they can open and close like stomata in plants. And that is where the gases can enter and leave. The trachea is a network of internal tubes. So once the gases enter through the spiracles, they then go through the trachea, and they do have rings within them to keep them open the whole time. And then the trachea branch into tracheoles, and these are much, much smaller tubes that are going to reach the abdomen of the insect. And they extend throughout all of the tissue in the insect to ensure that oxygen is delivered to all respiring cells. So those are the three key structures. Now, in terms of the adaptations, we need to think about large surface area, short diffusion distance, maintaining the concentration gradient. So first of all, tracheoles, which are the branching structure where gas exchange actually happens, have very thin walls. So that provides a short diffusion distance. Tracheoles are also highly branched, which provides a short diffusion distance because they're reaching so many cells that there's never going to be a big distance between the tracheoles and these cells. And also because the tracheoles are highly branched, that provides a large surface area for gas exchange. So these are our adaptations for short diffusion distance and large surface area. Linking to the maintaining of a concentration gradient, at rest, so when the insect isn't flying, diffusion is the main method of gas exchange happening at those tracheoles, and when the cells are respiring, they're using up oxygen and producing carbon dioxide. And that is enough to create a steep concentration gradient from the atmosphere compared to the tracheoles for the insect. So that is how the concentration gradient is maintained, is through respiration. And that's when they're at rest. During periods of high activity, so for example when they're flying, there will be mass transport of the gases occurring. So what happens in this case is the insect's abdomen will contract and relax, which actively moves air in and out of the tracheal system, and that helps to maintain the diffusion gradient. Finally, when the insect is very active, so they might be flying, they will end up respiring anaerobically. And lactate or lactic acid is produced during anaerobic respiration. And when that dissolves in the cells, it's going to lower the water potential of those muscle cells. And this causes water, which is naturally present in the very tips of the tracheoles, to move into the cells from the tracheoles by osmosis. And because water has now left the tracheoles and gone into the cells, there is now an empty space to be filled with air in the tracheoles. And that is going to cause gases to move more quickly into the tracheoles. Now, when it comes to reducing water loss, in the spec, it says you need to know how terrestrial insects reduce water loss and plants, and you need to know comparisons between the two. So let's have a look at terrestrial insects first of all. Insects have that waterproof exoskeleton, and that prevents evaporation of water across their surface. The spiracles that they have can open and close, and when they close, that prevents water from evaporating out. So that reduces water loss. And the spiracles also have tiny, tiny microscopic hairs around them as well, and that will trap any humid air, reducing the water potential gradient and therefore reducing evaporation. We'll come to the examples of the plants once we've gone through gas exchange in plants. So the structure or the cross-section of a leaf, we have at the very top the waxy cuticle, the upper epidermis, the palisade mesophyll, which is the site of photosynthesis, spongy mesophyll, that's where we have lots of air spaces. We then have the lower epidermis, and within the lower epidermis there are guard cells which create these pores known as stomata. This bit here is the vascular bundle, which is made up of the xylem and the phloem. And we can see those structures here under the microscope as well. So gas exchange in plants then, the palisade mesophyll, as we just said, that is the site of photosynthesis. So that is where lots of oxygen is going to be produced and carbon dioxide is going to be used up. That is what is creating the concentration gradient. So because we have that concentration gradient, oxygen will then travel through the air in those spaces in the spongy mesophyll and diffuse out of the stomata created by the guard cells, assuming that oxygen is not being used in respiration. Carbon dioxide, because that's being used up, we also have a concentration gradient compared to inside the leaf and the outside, so carbon dioxide will diffuse in through the stomata. So that is how the concentration gradient is maintained. To reduce water loss, the stomata will close when it is not light, so at night, because they won't be photosynthesizing, so they don't need carbon dioxide. The short diffusion distance is the fact that leaves are so thin, so it's a very short distance for the gases to diffuse from the outside in the atmosphere through the stomata to the palisade mesophyll. And the large surface area, leaves are very broad, so you're going to have lots of these cells and also lots of stomata, so there's a large surface area for that gas exchange as well. So the gas exchange at the stomata, oxygen diffuses out, carbon dioxide diffuses in, and we've just said to reduce the water loss, stomata close at night. Now, the other adaptations, in particular in xerophytic plants, which are plants that are adapted to survive in really hostile abiotic factors where there's very little water, the structural adaptations that they have are, first of all, a lot of the time the leaves curl up, and that means all of the stomata are facing inwards, and any water that does evaporate gets trapped, and it creates a very humid atmosphere to reduce the water potential gradient, so less water evaporates out. You also have these hair-like structures, which again trap humid air to increase the humidity. Sunken stomatas, again, that increases the local humidity. And you have a thicker waxy cuticle as well to help reduce evaporation too. You do have a longer root network. Now that's not going to reduce water loss, but that means you can reach more water to more readily replace the water that might be lost. Next then, 3.3.3, digestion and absorption. So during digestion, the definition of digestion is large biological molecules are hydrolyzed into smaller molecules that can be absorbed across cell membranes. And in mammals, we will learn about how carbohydrates, lipids, and proteins are digested. Those are the three biological molecules that you need to know. So first of all, the carbohydrates, they require more than one enzyme to hydrolyze them into the monosaccharides. It's amylases and membrane-bound disaccharidases. And the amylase is produced by both the pancreas and the salivary glands. So that's why digestion begins in the mouth, because the salivary glands will release amylase, and then in the mouth, any starch that is there will start to be digested. That will then be swallowed, goes into the stomach. The stomach acid will denature that amylase, and that is why the pancreas then releases more amylase into the very top part of the small intestines known as the duodenum, and you do have to say the duodenum to get the mark. And because we've now got more amylase being released there, any remaining starch can continue to be digested. Then as that food travels into the next part of the small intestines called the ileum, most of that starch should have now been digested into disaccharides. And embedded within the membrane of the epithelium of the ileum, you have membrane-bound disaccharidases, meaning enzymes embedded within that membrane that can digest the disaccharides into monosaccharides. So that could be the enzyme sucrase, digesting sucrose, or lactase, digesting lactose, or maltase, digesting maltose, all of which will hydrolyze the disaccharides into the monosaccharides, which are then small enough to be absorbed from the ileum into the bloodstream. So here are the three word equations for biological molecules, but just as a reminder here, because this could come up on paper 1, so that means even though this is paper 1 theory, it could come up linked to digestion, knowing what the 3 disaccharides are digested into or hydrolyzed into by those membrane-bound disaccaridases. Next then, proteins, and this starts in the stomach, it continues in the duodenum, and then it's fully digested in the ileum of the small intestines. And there's 3 enzymes involved in hydrolyzing these large polymers into the monomer amino acid. We have the endopeptidases. These hydrolyze the peptide bonds between amino acids in the middle of the polypeptide chain. That means that you're going to be breaking or hydrolyzing one long polypeptide chain into multiple shorter chains. And this increases the surface area for the next enzyme to work with. And that is the exopeptidases. And these hydrolyze peptide bonds between amino acids at the end of the chain. So by hydrolyzing the chain into many smaller ones, that's what the endopeptidases do, that increases the number of exposed ends for the exopeptidases to work on. So those two work together, doing the same thing, hydrolyzing peptide bonds, just in different locations to speed up protein digestion. Now, if there are any dipeptides remaining, meaning 2 amino acids still joined together, by the time that protein or digested protein then reaches the ileum, it's the membrane-bound dipeptidases, which are enzymes that can hydrolyze the peptide bond between 2 amino acids. Those enzymes will work to hydrolyze a dipeptide into amino acids, and the amino acids are small enough to be absorbed. So in terms of these proteins, you would have in the stomach the endo and exopeptidases. The pancreas will release more of those into the duodenum, and then in the ileum you have your membrane-bound dipeptidases. Next then is the lipids, and these are digested by only one enzyme, lipase, but also by the action of a chemical called bile salts. So lipase is produced by the pancreas and it releases it into the duodenum. And lipase can hydrolyze the ester bonds in the triglycerides. And that's the bond between the glycerol molecule and the fatty acids. So by hydrolyzing those ester bonds, you are hydrolyzing and releasing those fatty acids, and you do get monoglycerides as well. Bile salts are not enzymes, but they are produced by the liver. That will then be released into the gallbladder, and then the gallbladder releases it into the duodenum. And what bile salts do is emulsify these large lipid droplets into smaller droplets to increase the surface area for lipase to work on. They're also important in the formation of micelles. That comes up in lipid absorption rather than digestion. So this can be split down into the concept of physical and chemical digestion. The physical digestion is this emulsification. So the bile salts produced by the liver emulsify the lipids, meaning splitting it up into smaller droplets, but no chemical bonds are broken. So it's not chemical digestion, it's just splitting it up into smaller droplets so that there's faster hydrolysis by lipase. Chemical digestion is when bonds are broken, and lipase hydrolyzes the ester bonds so that we get glycerol, fatty acids, and monoglycerides. A monoglyceride is the glycerol molecule with one fatty acid still attached. Next then we move on to absorption, And in mammals, the products of digestion are absorbed across the cells lining the ileum, which is within the small intestines. So adaptations to increase absorption, and this is going back to this concept of this is an exchange surface, so we need to look for adaptations that increase the surface area, reduce the diffusion distance, and maintain the concentration gradient. So the ileum wall is covered in villi, which are these foldings, which have thin walls surrounded by a network of capillaries, and also these epithelial cells have even smaller microvilli, which is the folding of the cell membrane. And it's those features that maximize absorption. So the increased surface area is the villi and the microvilli, because you've got that highly folded surface. Decreasing that diffusion distance is because there's thin walls, but also the capillary network branches all the way up into the villi. So it's a short distance to diffuse across to reach the capillary network. Maintaining the concentration gradient is also because of that capillary network, because any digested food molecules that do get absorbed are transported away in the blood in the capillaries. Now, I've also got a table of adaptations listed here that I repeatedly have seen come up in the mark scheme. So you can screenshot this, turn it into a flashcard. These are what come up in long answer questions, short answer questions for adaptations to maximize absorption of digested products. So learn that. So now we've had a look at the adaptations for absorption, we need to know how the products of lipid digestion are absorbed, and also how the products of carbohydrate and protein digestion are absorbed. Starting with lipid absorption. And now we get to find out more about what are my cells. So my cells are water-soluble vesicles. And these vesicles contain fatty acids, glycerol, and monoglycerides. Those are the products of lipid digestion. So your triglyceride, after it's been digested, that's what we have. And it's also bile salts combined with those. And it's the bile salts that make these products, these fatty acids, water soluble. So that's what a micelle is, but a common exam question is, how do micelles help with lipid absorption? And they do it in these ways. So we've just said that the micelle is fatty acids, monoglycerides, bile salts, and those bile salts help to make the fatty acids soluble in water. Now that is an advantage for lipid absorption because it allows the micelles to move the fatty acids and monoglycerides from wherever in the lumen they were digested towards the cell surface membrane of the epithelial cell. Because the contents of the lumen, which is like the hollow space, the hollow tube of your small intestines, that is containing lots of watery substances from digestion. And fatty acids are not soluble in water. So we need them to be soluble so they can move easily towards the epithelial cell surface membrane. So that's why being water soluble is an advantage. So the second mark links to that. The micelles carry and then release the fatty acids and the monoglycerides to the epithelial lining of the ileum. So that then means the fatty acids are all released at this point. It's actually often another marking point from here that it helps to maintain the concentration gradient of the fatty acids near the epithelial cell. And the fatty acids, monoglycerides and glycerol, those are then absorbed by simple diffusion. The micelle does not enter, the micelle releases those at the cell lining. So those will then enter by simple diffusion because the cell membrane is made up of phospholipids and only non-polar molecules can diffuse through those, and fatty acids are non-polar. So inside of the cell, once it's been absorbed in the cell, it will then be reformed into triglycerides, and that happens in the Golgi body. So we get them packaged into these triglycerides, into vesicles, and those vesicles containing the packaged triglycerides will then move towards the cell surface membrane on the other side, fuses with the membrane, it then releases that triglyceride, which is picked up by a lacteal rather than the blood in the capillaries, and that is the end point of the absorption. Then we need to look at how the products of carbohydrate digestion and protein digestion are absorbed. So products of carbohydrate digestion are monosaccharides, for example, glucose. Products of protein digestion are the amino acids. And both of these are absorbed into the blood in the same way, and it is co-transport, which you learn in topic 2. So for co-transport of glucose, and it'd be exactly the same for amino acids, first of all, we have sodium ions from the epithelial cell actively transported into the blood. This creates a concentration gradient of sodium ions from the ileum into the epithelial cell. And therefore, the sodium ions can enter the epithelial cell down their concentration gradient, but they are an ion, so they have to go through a protein. And the protein that they can transport through is a co-transporter protein, which will bind sodium ions and glucose or amino acids. And that is then how the glucose and, if it was amino acids, enter the epithelial cell. Once inside of the epithelial cell, we already have a concentration gradient then. There'll be lots of glucose in the cell compared to in the capillary. And that means then that glucose can move down its concentration gradient into the blood and the capillary by facilitated diffusion through a transport protein. And it has to be facilitated because glucose is too large to move by simple diffusion through the phospholipid bilayer. Next then, 3.3.4 mass transport. Starting with hemoglobin, and these are groups of proteins found in different organisms. And hemoglobin is a protein with a quaternary structure. Quaternary structure from topic 1 means a protein with more than one polypeptide chain. And hemoglobin has 4 polypeptide chains, each of which has a heme group attached, which is where you have the iron ion. And it's this heme group and the ion where oxygen binds. So one hemoglobin molecule can bind 4 oxygen molecules, and then it transports the oxygen to respiring cells. Myoglobin sometimes comes up in application questions. So just to give you an awareness, this is another protein that can transport oxygen. It's not hemoglobin, but it's like hemoglobin in that it can transport oxygen, but it can only transport 1 oxygen molecule at a time. And some animals have a lot of this, and we have it in our muscle tissues. So oxyhemoglobin dissociation curves. Oxygen is going to be loaded in areas in the body with a high partial pressure of oxygen. So for example, the alveoli in the lungs, and it will be unloaded in regions of low partial pressure, so where oxygen's been used up for respiration. So on our oxyhemoglobin dissociation curve, this axis is showing us the percentage saturation of oxygen on the hemoglobin at different partial pressures of oxygen. And that basically means the amount of oxygen present. Partial pressure is a way to consider concentration. So in areas where there's a low partial pressure, meaning not a lot of oxygen present, that will be respiring tissues because oxygen's constantly used up in aerobic respiration. And at areas with a higher partial pressure, for example, the alveoli in the lungs, that is because as you inhale, you're getting new air, which will have lots of oxygen within it. So this oxygen dissociation curve, or oxyhemoglobin dissociation curve, it shows us that at low partial pressures, hemoglobin is not as saturated as it is at high partial pressures. And if it's not as saturated, that must mean it's either unloading oxygen or not loading oxygen as much. And we'd say at that point, that would mean the hemoglobin has a low affinity for oxygen. Affinity being the attraction and ability to bind oxygen. Whereas at higher partial pressures, because the hemoglobin is nearly fully saturated with oxygen, that must mean that hemoglobin has a very high affinity for oxygen. It's loading lots of oxygen onto it. Cooperative binding is this concept of when you have different numbers of oxygen bound to the hemoglobin, it affects the ability of subsequent oxygen molecules to bind. So hemoglobin can associate with, which means load, 4 oxygen molecules because of those 4 heme groups. And as each molecule binds, it slightly changes the shape of hemoglobin, exposing the further binding sites, making it easier for each subsequent oxygen to bind. So if we think of this as, we've got 0, let's say that's the first one binding, second oxygen binding, third and fourth. We can see that we've got not a particularly steep gradient compared to the point of 2 and 3, showing that it must be hardest for the first oxygen to bind. And that's because at this point the binding sites are slightly covered. Once that first oxygen binds, it changes the shape and exposes the binding sites. So we can see we've now got a steeper gradient, and that is representing that it's easier for the second and the third oxygen to bind. However, when we get to the fourth oxygen, we can see the gradient is less steep again, and then it plateaus. And that's because if 3 of the 4 binding sites are already saturated, you've got a 1 in 4 chance now of the oxygen binding with an empty binding site. And that's why the ability to bind the fourth one is also more unlikely or challenging. So this all shows us that in areas with a high partial pressure of oxygen, as we said, there'll be a higher affinity. In areas with a lower partial pressure, there'll be a lower affinity. The Bohr effect is looking at the effect of carbon dioxide dissolved in the blood on hemoglobin's affinity. And when carbon dioxide dissolves in the blood, and by the way, this carbon dioxide is coming from aerobic respiration. So when you're respiring lots aerobically, you will have a lot of carbon dioxide dissolving in your blood, forming carbonic acid. Carbonic acid decreases the pH of the blood, and that decrease in pH slightly changes the tertiary structure of the polypeptide chains in hemoglobin. And this is why the affinity for oxygen decreases in respiring tissues. That change in shape is going to decrease the affinity. So hemoglobin ends up loading less oxygen and unloading more. And that is an advantage, because if hemoglobin is going to be unloading oxygen more readily at respiring tissues, that means it's delivering the oxygen to the site of aerobic respiration where it's needed, so aerobic respiration can continue. And the way we can see this on the graph is, as the pH becomes more acidic, we can see the curve shifts to the right each time. And the best way to consider this in an exam is, at any of the partial pressures, use your ruler, which I don't have right now, and draw a vertical line and read off what the partial pressure is. Sorry, the saturation, not the partial pressure. So read off what the saturation is for that partial pressure. And let's say this one is maybe 25, 39, and that might be 56. That's very inaccurate readings off. But the point is, even at the same partial pressure, the most acidic is 25% saturated. The least acidic is 56% saturated. So what this shows us is the hemoglobin in the more acidic conditions must have a lower affinity, indicated by the fact it's less saturated, so it's more readily unloading oxygen rather than loading. So if a curve shifts to the right, that is what it's indicating, and this is how I demonstrate it in an exam. Read it off and show that even at the same partial pressure, the saturation is lower, showing that it must have a lower affinity for oxygen. Within the spec, it does say you can get applications linked to different hemoglobins for animals. So it says you need to know that animals are adapted to their environment by possessing different types of hemoglobin with different oxygen transport properties, which basically means, based on their environment, they've evolved to have hemoglobin with either a higher affinity for oxygen if they live in an environment which has a low partial pressure of oxygen, or they might have evolved to have hemoglobin with a lower affinity for oxygen if they need to readily unload it for constant aerobic respiration at higher rates. So things like small organisms that need to maintain their body temperature and they lose more heat across their surface. You might also have questions linked to fetal hemoglobin and myoglobin as well. Myoglobin has a very high affinity for oxygen, even at very, very low partial pressures, and in that way it can act as an oxygen store. Linked to the fetal hemoglobin, this is showing us in blue the curve for fetal hemoglobin, and green, the adult hemoglobin, and that would be the mother, for example. And a fetus doesn't breathe. It gets all of its oxygen from the placenta as the blood then goes from the placenta, passes across the umbilical cord and into them. And that means that it's really important that a fetus has hemoglobin with a higher affinity for oxygen than the mother's hemoglobin's affinity for oxygen. Because that then will show us, again, I'd draw a line. Let's say we were at 25 partial pressure. We can see that we're maybe 45% saturated, whereas here we're 70%. So that would mean at this partial pressure, some of the oxygen is being, almost half of the oxygen, in fact over half of the oxygen has been unloaded from the mother's hemoglobin. And that can then be picked up by the fetus's hemoglobin because they are still at a high saturation, indicating a higher affinity. So at these lower partial pressures, the adult, the mother hemoglobin, will be unloading oxygen, but the fetus's hemoglobin, which has a higher affinity even at low partial pressures, will be able to load up the oxygen still. And in that way, a fetus gets oxygen from the mother. So we move on to the circulatory system, and mammals have a closed double circulatory system. Closed meaning the blood remains within the blood vessels and the heart, whereas the double circulatory system means the blood passes through the heart twice in each circuit. So one circuit goes to the lungs, one circuit goes to the rest of the body. And mammals require a double circulatory system to help manage the pressure of the blood flow. So the lungs are directly behind the heart. So they're not a very far distance at all. So you don't need a high blood pressure to make sure that the blood reaches the lungs. And in fact, we don't want a high blood pressure because that could damage the capillaries in the lungs if the blood was pumping through at that high pressure. And also it's an advantage to have a lower pressure because that reduces the speed at which the blood flows, which means there's more time for gas exchange in the alveoli. In contrast, we do want a high blood pressure for the blood flowing in the circuit from the heart to the rest of the body because we need it to ensure it can reach all of the respiring cells in the body. You need to know some key blood vessels. It's not all of the ones in the body that we can see here, but it's the ones in the red boxes. So you need to know the coronary arteries, which we can't see here, but these are the blood vessels that are on the outside of the cardiac muscle on the heart, and it supplies the cardiac muscle itself with oxygen for respiration. And then you need to know the blood vessels that are connected to the heart, the lungs, and the kidney. So we'll start at the top of the lungs. Any blood vessel that is connected will have pulmonary if it's for the lungs. And artery means it's blood coming away from the heart. Vein, if you think in, it's going back into the heart or towards the heart. So pulmonary artery is blood coming away from the heart to the lungs. And pulmonary vein is the blood vessel that is taking blood from the lungs back into the heart. Then if we move on to the heart, we've actually covered two already because the two that are connected to the lungs are also two connected to the heart. So then if we have a look at the blood vessels carrying blood away from and into on this side, the aorta is the artery carrying blood away from the heart to the rest of the body. The vena cava is the vein carrying blood from the body back into the heart. Vena cava meaning body vein. And lastly, the kidneys. Any of the blood vessels that are attached to the kidneys have renal in front of them. So renal vein means it's the blood vessel from the kidneys taking blood back into or towards the heart. And renal artery means it's blood coming away from the heart to the kidneys. So those major blood vessels are then connected within the circulatory system via arteries, arterioles, capillaries, also venules, but you don't need to know that for AQA, and veins. And that takes us on to these blood vessels. So arteries in general, away, they carry blood away from the heart towards the arterioles. Arterioles are smaller than arteries, they're branching and they connect to the capillaries. You don't need to know about the venules, but you then have the capillary network, and then you learn about the veins, which carry blood towards the heart. And I always say like vein, it's got in it, it's going back in or towards the heart, away. So we can see this here just from another angle, and it's showing us the inside of the walls, which are the adaptations that you need to know for the blood vessels. Now, the aorta is the largest artery in the body. It carries blood at the very highest pressure in the body. And it carries it at high pressure because it's carrying it directly from the heart, where that left ventricle would have just contracted, forcing the blood out at high pressure. So it contains a large amount of elastic tissue in its wall, which means that it can stretch when the ventricle contracts, and it can recoil when the blood is then flowing away, causing it to move back to its original shape. And this helps to smooth out blood flow and also maintain consistent pressure. It's got a very thick muscular wall as well, and that helps it to withstand high blood pressure and prevent it from bursting, whilst also the muscular wall can contract, and that helps to smooth the blood flow as well. Arteries in general, aorta is an artery, but it's the thickest or it's the biggest. Arteries all have thick muscular and elastic walls. That means they've got a narrow lumen for the blood to flow through, and it helps them to maintain the high blood pressure as they are directing the blood from the heart away to all of the cells of the body. Arterioles are going to be branching from the arteries and help to regulate blood flow into the capillaries. So they still have a relatively thick layer of muscle, and that can contract to narrow the lumen or vasoconstriction. That reduces the blood flowing into the capillaries. And when the muscle relaxes, the lumen widens, which is vasodilation, increasing the blood flow to the tissues. Lastly, in the veins, these carry blood back to the heart under low pressure. They have thinner walls. They still have muscle and elastic within the walls, but just a much thinner layer. And because it's a thinner wall, they have a wider lumen. And this is to reduce resistance of the flow. And that's because the blood is going to be flowing at a much lower pressure because it's now further away from the heart, which was the pump that increased the pressure in the first place. It also has valves which prevent the backflow of blood. So here's a little summary table of some of those features that you can screenshot and add to your notes. Capillaries then. These are the smallest of the blood vessels and adapted for efficient gas exchange or other exchange of substances between the blood and surrounding tissues. And some of the adaptations are, first of all, very narrow diameter, which means only one red blood cell can fit through at a time. That slows down the flow of blood, which is good because that means there's more time for exchange, for example, gas exchange. The walls are also only one cell thick, so it's consisting of a single layer of flattened endothelial cells, and that reduces the diffusion distance. Capillary beds are networks of capillaries that spread through tissues, and that provides a large surface area for exchange as well. And the extensive branching also means that all cells are going to be close to a capillary, and that is reducing the diffusion distance. So again, we're thinking here about large surface area and reducing diffusion distance. This leads us into tissue fluid. Tissue fluid is a fluid surrounding your tissues that contains water, glucose, amino acids, fatty acids, ions, oxygen that have all been forced out of the blood in the capillaries. So you need to know, how is tissue fluid formed, and then how does the water from the tissue fluid get reabsorbed? So in terms of the formation, the capillaries, we've said that it's only a single layer of cells making up that endothelium wall, that lining. And between every cell, there's going to be these microscopic gaps. So any molecule that's small enough to fit through those gaps can be forced out if there's a high enough pressure. And that's what we have at the arterial end. So this is where we'd have arteries, that branches into arterioles, and then that branches into the capillaries. And at the other end, the capillaries, then there's the venules and then the vein. At the arterial end, there is a high pressure. And that high pressure is originally generated from the contraction of that left ventricle in the heart. And then also the arterioles have got a wider diameter than the capillaries. So going from that larger diameter to a smaller diameter with the same volume of blood increases the pressure. And we call this high hydrostatic pressure. And because there's such high pressure, anything that's small enough to fit through those gaps between the cells and the capillaries is forced out, and that's known as ultrafiltration. Now, the key thing here to get the mark, you would have to name what gets forced out, not just say small molecules. So always name water, and then I would name at least 2 other things from this list. So water, glucose, amino acids are forced out. That is how tissue fluid is formed. So what is forced out then? Water, dissolved mineral ions, glucose, small proteins can, amino acids, fatty acids, and oxygen. But anything that's too big to be forced out will remain in the blood in the capillaries. And that is primarily large proteins, also red blood cells and platelets, but the marking point is normally large proteins. And this is important because it helps to explain how the water from tissue fluid gets reabsorbed. So because those proteins remain in the blood in the capillaries, you end up with a much lower water potential at the venial end of your capillary compared to the water potential of the tissue fluid. And that is how water can then move from the tissue fluid back into the capillaries by osmosis. Now, in terms of mark scheme language, you must say it's water that is re-entering, not tissue fluid. So it's water that is entering by osmosis because of this decreased water potential. Now, within that water, there might be dissolved substances that are also reabsorbed, but that's not what you need to say. Because this is osmosis, that means eventually you will reach equilibrium in the water potential because water is moving in by osmosis. And when equilibrium is reached, no more water from that tissue fluid can be reabsorbed. But we need to reabsorb as much of it as possible, otherwise you get swellings in the body where you've got too much water outside of the blood vessels. And that's where the lymphatic system absorbs the rest. So then we move on to the heart, starting with the cardiac muscle that it is made from. So you get these thick muscular layers of cardiac muscle, and that has unique properties such as it's myogenic, which means it can contract and relax without any input from the nervous system or hormonal stimulation. So as long as it has a constant supply of glucose and oxygen in the blood in the coronary arteries, it can contract and relax. So it never fatigues as long as it does have that oxygen and glucose for aerobic respiration to make ATP so it can contract. So that's then our coronary arteries and the cardiac muscle. And this is just showing us the coronary arteries in more detail. So these branch off from the aorta, carrying oxygenated blood and also glucose that's needed for aerobic respiration. If one was to get blocked, this is now actually application, it's not on the spec anymore, but if you did have a blockage in your coronary artery, that means that all of these blood vessels would not have oxygenated blood flowing through them anymore. So all of that cardiac muscle would not get oxygen, glucose, wouldn't be able to respire, and it would stop contracting. And that is what a heart attack is, or myocardial infarction. It's when the heart, cardiac muscle stops contracting. In terms of the structure of the heart, you have 4 chambers. So you have the 2 atria at the top, the 2 ventricles at the bottom. The atria have much thinner muscular walls, and that's because they don't need to contract with much force at all, because they're only moving the blood from the atria directly down into the ventricles. And they're elastic walls to stretch when the blood enters. Ventricles have much thicker cardiac muscle, and that's so that they can contract with more force because they're pumping the blood further. The right ventricle is pumping blood to the lungs. The lungs are directly behind the heart, so it doesn't have to pump a very far distance. They don't need as thick a muscular wall compared to the left ventricle, which does have to contract with a large force to create a high blood pressure to pump the blood to all of the respiring cells in the body. So the left ventricle wall has the thickest cardiac muscle. Next is knowing the different blood vessels, which we actually touched on already because we talked about the pulmonary arteries are carrying blood away from the heart to the lungs. Pulmonary veins carry blood from the lungs back into the heart. The aorta is the artery that carries blood from the left ventricle to the rest of the body. And the vena cava carries blood from the body back into the heart. Next is looking at the valves in the heart. So the semi-lunar valves are the ones between the ventricles and the arteries. So you have one in the aorta, which you can't actually see in this diagram, but it's just about here. And then we've got one here in the pulmonary artery, just there. The atrioventricular valves are found between the atria and the ventricles, and we have them both on the left and the right. The bicuspid is the name for the one on the left side, and the tricuspid is the name for the one on the right, but it's sufficient just to call them both atrioventricular for AQA. Now, valves will open when the pressure is higher behind them, forcing them open, and a valve will close when the pressure is higher in front of them. And in this way, they control the flow of blood so that it only goes in one direction, and it prevents the backflow of blood in veins, but also within the heart, making sure that the blood does go from the atria into the ventricles, out of the arteries. The septum, now this isn't technically on the spec, but just to make you aware, the septum is the part of the cardiac muscle that runs down the middle, separating the left and the right side, meaning the oxygenated blood from the deoxygenated blood, and that is going to help maintain concentration gradients for diffusion, because you're going to have far higher concentration of oxygen being pumped around, whereas if this all mixed together, it's going to decrease the concentration of oxygen in the blood. We then move on to the cardiac cycle, and there are three stages: diastole or diastole, atrial systole or systole, ventricular systole or systole. People pronounce it differently, so I'm going to keep switching between the two because I haven't decided how I'd pronounce it yet. So if we start with diastole, this is relaxation, and diastole is when both the atria and ventricle cardiac muscle are relaxed, and all of this pale green bit is our ventricular diastole, and all of this section is atrial diastole. But diastole means the cardiac muscles are relaxed. And this is when the blood is going to be entering from the body into the atria, when we've got both the atria and the ventricles in diastole. And it's going to enter through the vena cava and the pulmonary vein into the right and the left atria. The blood flowing into the atria is going to increase the pressure within the atria compared to the ventricles, and that causes the atrial ventricular valves to open, and so some blood will start to move into the ventricles at this point. Atrial systole, wherever you see systole, it means contract. Atrial systole is a very, very brief period. It's not a long stage, and this is when the muscles in the walls of the atria contract, increasing the pressure further, forcing all of the remaining blood that was within the atria into the ventricles. The ventricular muscle walls are relaxed at this point. We can see they're still in diastole. Then we have ventricular systole. So this is when the ventricles contract. And this happens after a short delay, meaning the atria contract, there's a short delay, and then the ventricles contract. And as they contract, that's going to increase the pressure in the ventricles to the point where the pressure is now higher in the ventricles than in the atria. And that is what causes the AV, or the atrioventricular valves, to close. And then when the pressure is even higher compared to the arteries, it causes the semilunar valves to open. And that is what causes the blood to be pushed out of the ventricles through the two arteries, either to the lungs or to the rest of the body through the aorta. Now, there is a math skill linked to this, the cardiac output, and that is the volume of blood which leaves one ventricle in 1 minute. And it can be calculated by doing the heart rate times the stroke volume. And heart rate is beats of the heart per minute. Stroke volume is the volume of blood that leaves the heart each beat. The final part is mass transport in plants. So we're going to look at transpiration first, the mass transport of water. And transpiration is the loss of water from the stomata by evaporation. And there are 4 key factors that can affect the rate of transpiration. Light is one of them, because the brighter it is, the more the stomata open. So there's a larger surface area for that evaporation or transpiration of water. Temperature, if you've got a higher temperature, there's more kinetic energy, so the water molecules will move faster. So again, more evaporation and transpiration. Humidity, if it's more humid, there'll be less transpiration. And that's because the more humid it is, the more water vapor in the air, and that will reduce the water potential gradient, so you get less water evaporating. The windier it is, that's going to blow away humid air containing that water vapor, and that can help to maintain the water potential gradient, so increasing that rate of transpiration. Then if we have a look at the movement of water up the xylem, water has to move from the roots all the way up a plant against gravity. And sometimes it's a small plant, sometimes it's a plant that might be several meters tall, moving water against gravity. And this is possible because of the cohesion tension theory. And that's what we're going to have a look at next. What cohesion is, what capillarity or adhesion is, and what we mean by the tension part in this theory. Now this links back to module 1 with the properties of water. Water is a polar molecule, meaning it's got a slight negative charge on the oxygen atom, slight positive charge on the 2 hydrogen atoms. And for that reason, hydrogen bonds can form between the oxygen and hydrogen of different water molecules. That is cohesion, those hydrogen bonds between water molecules sticking the water together. And that means that water can move up the xylem as a continuous column of water. Now you also get hydrogen bonds forming between the water molecules and the walls of the xylem, and that is adhesion, the sticking of those water molecules and then the entire water column to the walls of the xylem. And that can result in the xylem being drawn in more and more narrow, because we're going to look at how the water column gets pulled. And if you're pulling upwards on that column and the column is all attached and it's stuck to the wall, as you pull it up, it's like an elastic band. It's going to cause the xylem to become longer and narrower. And the reason that helps move water up against gravity is the narrower the tube, the more surface area you have for the water column to stick to, and that means it can actually move up the tube with more ease, which is just shown here with like different diameter straws. So the cohesion tension theory then. Water evaporates from the stomata. That is transpiration. That would be your first mark. Water evaporates from the stomata. As that water evaporates, it decreases the water potential in those surrounding cells, and that creates tension in the water column and on the xylem, and that draws and it pulls the water up. So that'd be your next mark. As that water is lost by transpiration, that creates tension on the water column and it pulls the water column up. The water molecules stick together because of cohesion. So that's your third mark. All of those water molecules form a continuous water column because they stick together because of the cohesion by the hydrogen bonds. And then at the same time, we do also have the column of water sticking to the walls of the xylem, adhesion, and that helps to move it up and it also draws in the xylem narrower under that tension. So that is our cohesion tension theory. As I've read this through, this is a lot of text here. That's to give you the full understanding, but the bits that are in bold, those are your key marking points typically, and the ones that I pointed out as well. So then we have a look at the mass transport of sucrose in plants. And this is one of our products of photosynthesis. And all cells need this sucrose to then break down or hydrolyze into glucose for respiration. So this is going to be transported from the leaves, where the organic substances are made in photosynthesis, to all the respiring cells through the phloem. And the sugary solution, that sucrose solution, can move up or down through the phloem, whereas in the xylem, it only ever moves upwards. So the phloem tissue, there are two key cell types. We have the sieve tube elements and the companion cells. The sieve tube elements are here in the middle. They are living cells, but most of the organelles have disintegrated. So there's no nucleus and they don't have many other organelles. And that's why they need these cells next to them, called companion cells. And the companion cells will have lots of mitochondria in them so that they can do lots of aerobic respiration and provide the ATP needed for active transport in this process. So this links to the mass flow hypothesis, and this is looking at how the sucrose that's made in the leaves, which is our source, can be transported to the respiring cells, which is known as the sink. So we're going to go through this process of how it happens. There's a lot of text here, which I'm not going to talk through at this point. By all means, you can pause, write it down, but it's also going to come up in the subsequent slides. So I'm going to talk through it with those. This has just got a lot of the information in one place for you to screenshot if you want. So the source to sink explanation with this model helps to explain the mass flow hypothesis in terms of how the hydrostatic pressure causes the mass movement of sucrose solution. So in this tank, this model, we've got a tank of water. We've got one tube representing the phloem, another tube representing the xylem. And our source cell would be our photosynthesizing cell, where you'd have lots of sucrose being produced. And sucrose lowers the water potential of the cell and also of the phloem. And that means water is going to enter from the xylem into the phloem. At the sink cell, because lots of respiration is occurring, that's going to be using up sucrose, and that means you will now have a more positive water potential or less negative. So water will actually move from the phloem in this region back into the xylem, and that will decrease the pressure. So at this end, we have an increase in the hydrostatic pressure as water moves in by osmosis. And at this end, we have a decrease in hydrostatic pressure because water leaves by osmosis. And as a result, because we've got high pressure and low pressure, that forces the liquid through the phloem from the source to the sink. So you also need to know, well, what is it that is creating this hydrostatic pressure, and how does the sucrose move? So our source cell, we're looking at it slightly differently here now because it's actually in the plant rather than a model. The source cell is the leaf. That's where photosynthesis happens, sucrose is produced. Sucrose is actively transported from the source cell into the sieve tube element. So it has to go from the source to the companion cell to the sieve tube element. And that is done using ATP because it's active transport. And the ATP is provided by the companion cells. So this is your mark number 1 in translocation. Sucrose is actively transported using ATP from the companion cells. Now we've got the sucrose into the sieve tube element. That lowers the water potential of the sieve tube element, and therefore water from the xylem will enter the sieve tube element by osmosis. So here would be our mark 2. That increases the hydrostatic pressure. That would often be mark number 3, or these 2 points will be split between 2 and 3 marks. And because we've got a high hydrostatic pressure, that forces that sucrose solution to move en masse, so this is our mass transport, towards the sink cell. And that is because at the sink cell, sucrose is being used up in respiration, and therefore we're going to have sucrose from the phloem being transported into the sink cell, initially by facilitated diffusion, but when the concentration gradient is equal, you have equilibrium, you'll then have active transport of the sucrose from the sieve tube element into the sink cell. But either way, that means you have less sucrose here. That now means you have an increase in the water potential gradient, and water will move from the sieve tube element by osmosis back into the xylem. So it's that pressure gradient, the high hydrostatic pressure compared to a low hydrostatic pressure, because water enters by osmosis, water leaves by osmosis. That's what causes the mass flow, the mass transport of the sucrose solution from the source to the sink. Now, there are investigations that have been linked to translocation and working out the involvement of the phloem, which are tracers and ringing experiments. Tracers involves radioactively labeling carbon, and then that carbon is provided to the plant in the form of carbon dioxide. So by only providing the plant with radioactive carbon in the carbon dioxide, that means over time, all of the carbon-containing compounds in the plant should contain the radioactive carbon, because the carbon dioxide is taken in for photosynthesis and turned into sucrose, other sugars. So that means that you can take thin slices through the stem, and under the X-ray film, any radioactive carbon is going to show up. So in this way, you can track the route that the radioactive carbon takes through the plant, and that is how they could see where the phloem was, and that it must be the phloem that is transporting the sugars, because they are what lit up with that radioactivity under the X-ray. Ringing experiments is when a ring of bark, which will contain the phloem, are peeled and removed from a tree trunk. This can actually also be done on stems of smaller plants as well. And the result of removing the phloem is the tree starts to bulge here and here, so above where it's been removed. And that bulging, you can remove the liquid and analyse it to see it contains sucrose. But what this indicates is if you remove the phloem, the sucrose is made in the leaves further up, and when you take the phloem out, you get this bulging of liquid. The liquid contains sucrose, and that shows us that it must be the phloem transporting sugars, because when you remove them, the transport stops. So that takes us to the end of topic 3. And don't forget, if you do want more help with mark scheme specific answers, with all of the theory, examiner tips, fact recall questions as well, check out my notes. And if you just want to test yourself on all of the key marking points, then give my flashcards a go. But that is it for this video. I'll see you in a video very soon.